My research is on plants, and I've always been interested in how plant cells respond to the environment. My current research examines how contact sites between plant organelles and the endoplasmic reticulum facilitate molecular movement. These contact sites act as bridges, allowing molecules like calcium to transfer between organelles. The transfer impacts cellular signaling, which plays a vital role in plant adaptation to environmental change, growth, and in maintaining health. We also study essential molecules for building cell membranes called sterols. These sterols are involved in signaling and transport within the cell, and they also contain the important structural elements to provide membrane structure. Understanding their pathways can shed light on how plants regulate their cells and adapt to environmental stress, potentially revealing new strategies for improving plant resilience. One direct application of this research is the development of a non-toxic, organic, pre-emergent herbicide based on the sterol work. This herbicide is more cost-effective and more efficient than conventional options. It supports sustainable agriculture, including the cultivation of organic plants critical for producing medicines, such as opiates for pain relief and alkaloids for cancer treatments. More broadly, plant cell research helps us develop crops that are resilient to climate change and capable of enhanced nutritional value. As the global population grows, these innovations are critical for ensuring food security and meeting future demands. Ultimately, human survival is deeply tied to the health and productivity of plants. At Texas A&M University, we are fortunate to have a Nikon Center of Excellence and a Microscopy and Imaging Center, where researchers and students regularly use Nikon microscopes. The Nikon AXR confocal microscope, with its high-speed, high-resolution, and wide field of view feature, enables us to observe calcium oscillations in plant cells, revealing similarities with animal cells, like nerves and muscles. This insight highlights shared mechanisms between plants and animals, deepening our understanding of how plants respond to the environment and adapt to climate change. The AXR multiphoton microscope allows us to photostimulate plant cells, enabling precise studies of cell responses and molecular interactions at high depth and resolution. These tools make it possible to uncover details about plant cells that are crucial for advancing agricultural innovation and environmental research. Our work on sterols aims to uncover how these molecules move and are regulated within plants, an area that remains largely unexplored. With Nikon microscopes, we hope to further analyze these processes, which could help improve crop resilience and stress tolerance. Plants lack nerves, but possess ER systems that are remarkably resilient to stress. By studying these mechanisms, we may not only develop sustainable agricultural practices, but also discover ways to address human diseases related to ER stress, such as Alzheimer's and Parkinson's. With Nikon microscopes, we anticipate diving even deeper into plant cells, enabling more precise discoveries that can benefit both agriculture and medicine.